Thank you for joining the Museomics workshop of the DFG Priority Program 1991 Taxonomics. It's my pleasure to introduce you to the topic with the first lecture, basically introducing you from the start of the DNA extraction, and I'm looking forward to the other talks of the subsequent approaches to using herbarium material and museum material in taxonomic work. I have to admit that my title is fairly lame, but what I should have called it is actually how to get that damn DNA out of the stupid old plant to get it into my phylogeny. I have now more than 20 years experience with herbarium specimen work, and this can sometimes be really hard to get the DNA in a workable fashion out of a plant. So before you start, there are two things at least to consider. First, DNA is not conserved in a herbarium specimen. It is evolving, kind of. It fragments, mostly by depurination. It gets degraded and deaminated, which means cytosines are changed to uracil, which is then subsequently read as thymine. So you actually observe a C to T transition in old material. Second, DNA extraction means a mix-up between compartments of cells, no matter whether it's fresh material or herbarium material. In the end, it means that you mix things which are in the vacuole with the cytoplasm and the DNA of the nucleus. But in living plants, there is a reason why compounds of the vacuole, such as phenolic compounds and polysaccharides, are in the vacuole and stored away from the DNA in the nucleus. So before you start, you need to consider that the DNA is not really in the way that it is in the living organism, and you need to consider these in subsequent steps. So before you start, you need a clean room. This is especially important for working with herbarium material, which has only poor DNA. So it's important to separate the DNA extraction from other molecular work, and if possible, to actually separate DNA extraction from herbarium material from DNA extraction of fresh material. I admit that may not always be possible. But it is also important to have extra clean tables. Use bleach, ethanol, or UV light. Make sure windows are closed. Yes, I had a day when the windows were open and the whole group was only extracting maize DNA because the pollen flew in on the day when we were actually trying to extract DNA from herbarium material. Another recommendation is work on tin foil. Then make sure you clean the material you work with, such as pipette tips that needs to be autoclaved, or any kind of scalpel knife that you work with. Make sure you use filter tips to avoid contamination from within the pipette. Make sure you wear clean clothes, and make sure that you cannot contaminate your probes. So work with gloves, use long gloves, use a face mask, not a big problem at the moment. And in case of you have really problems with a precious herbarium material, better also use shoe overalls or a hairnet. So starting with the material. Herbarium material is not herbarium material. There is so much difference, and the most important is taxonomy. So if a DNA extraction protocol works for one taxon, it does not need to work in another one. There are so many different ingredients, such as phenolics and polysaccharides. Cates et al. Just published a study comparing the DNA extraction of 8,000 different specimens, and you can see in the tables below that the DNA yield on the left side and the proportion of reads on target reads differ enormously between different families of living plants. So, part of the problem is not just taxonomy, but also the way that material was preserved in the field before it got into the herbarium. And not all the people put it in the press only, but they tried to dry the material in other ways which may not be suitable for DNA extraction. A study by Sue Forrest demonstrated this really nicely, and she compared artificially made herbarium specimens in different ways. So she compared alcohol and drying room material with those just put into the press with material put in herbarium press and then dried using a hairdryer, material put into a drying oven, pickled material, so this is stored at room temperature in alcohol for 3 weeks, material put into RNA later, and of course favorite for most of us, as material put in silica gel. Her analyses really prove that many methods work, and even just putting into the herbarium press may work really nice. But important for you is, of course, material stored in alcohol does not work for DNA extraction, and material dried with a hairdryer does not work anymore. So this is heated too much and the DNA got destroyed. So even if your material looks nice, is young, it may not work for DNA extraction because it was dried too hot. Okay, and subsequently, also the preservation in the herbarium may not favor the DNA extraction afterwards. Pest control is an important part of conserving plants in the herbarium, and there are a multitude of different toxic materials that are put into different herbaria in a routine way in the past, but also nowadays, that damage DNA. So, in the previously mentioned study by Cates, the herbarium source was also a good predictor for DNA yield. But they found one hint which may be important for you. The greenness of material was an important predictor. So, if a plant was old but still green, DNA extraction worked. If it was brown and young, it may not work. So the first step in DNA extraction of plant material, and zoologists have it better in that way, is cell breakup. You need to make the plant into a fine powder. And there are many ways in which to do this. You can use the old-fashioned way with pestle and mortar in liquid nitrogen or fine sand. But you can also use machines like the Retsch mill, in which steel and glass beads are used to smash the dry plant material, or there are special tissue homogenizers. Whatever you do, the aim is to increase the surface from which to extract DNA. Once you have the fine powder, so the next step is the lysis, in which you dissolve the cellular proteins and the free DNA in your buffer. And you can do a lot of different things with the buffer. The most commonly used buffer is a CTAP buffer, which works at around 70 degrees for a specific amount of time. But there are many ways in which you can modify this step, especially the Lysis-buffer. CTAP is a chemical used to bind polysaccharides, proteins, and cell wall parts. Then you need salt in there to inactivate enzymes, EDTA to form halides, especially complexes with magnesium ions, which are needed by the plant for DNases. So with that, you prevent further degradation. Special care needs to be taken with some material that is especially gluey because of the many sugar in the plant material, which form a mucilage. Especially the succulent Cactaceae and Crassulaceae are known for that, but also Malvaceae and Poaceae. The most commonly used method for that is to add sorbitol and a high salt concentration, but there are also more specialized methods to deal with this. The Lysis-buffer further may contain PVP, which is a substance to bind phenolic substances, but it can also be used for latex-rich tissue. Other material chemicals used in there are, for example, beta-mercaptoethanol, which prevents further oxidation of DNA by different compounds, and RNAs may be in there. Then you can also modify the length of incubation time, and there's a lot of discussion in there. We have tested different times of incubation because in the literature it is said that you should use longer incubation times to increase yield. Yet in our experiments we found the opposite to that, with a decreased yield. And we found also an increasing amount of phenolic compounds with a longer incubation time. So you should be careful with increasing the incubation time. And there are also other possible modifications for the incubation, for example, the use of proteinase K. So once you have your buffer with the DNA, you can either use the Miniprep or you can use silicon membrane columns. The Miniprep follows the lysis with a chloroform precipitation step, which falls the proteins and cell wall material, and you have an upper aqueous phase with DNA and a lower phase with chlorophyll and proteins. And subsequently, you precipitate your DNA with alcohol. Most of you are likely familiar with the silicon membrane columns, which are an alternative to this chloroform cleaning, which basically works by binding DNA on silica membranes in a small tube at high salt concentration and low pH. And then you have washing steps, and then afterwards you dissolute DNA at low salt concentration and a high pH. There are many different columns out there on the market, and basically every major molecular biology distributor has its own kind of columns. It is worth comparing them. Our own experience, for example, comparing the Miniprep versus the DNA Easy Kit demonstrated that the Miniprep is better for herbarium material than the Easy Kit. One paper which will be mentioned afterwards is on butterflies, showing that the Monarch kit by New England Biolabs is the best one, which has a higher amount of small DNA fragments, which may be due to the extra ethanol in the cleanup of this kit, which basically is what also was found by Latoro et al., which suggested to add a step with binding buffer consisting of 3 molar sodium acetate, guanidine hydrochloride, and isopropyl alcohol to extract extra short DNA fragments which are common in herbarium material. So, what are the advantages of one over the other? The Miniprep is considered to be more flexible and to have a higher yield, although that needs to be tested more rigorously. The columns are more favored by those who want a faster one and less hazardous chemicals, and they have been shown to include less impurities afterwards. So let's check whether the DNA yield is really better in the Miniprep with a silica membrane. In our experiments with Veronica, this was not the case. In contrast, silica membranes seem to be slightly better for older material. But the yield was actually about the same for both methods. However, the yield depends less on age than rather the quality of herbarization. So supporting the notion by Cates before that the source, the herbarium source, is an important factor. But is DNA yield really important? In the analysis by Cates, yes, there was a slight positive relation, but this positive relation between yield and the proportion of target reads in the HYPE-SEQ experiment was not significant. So maybe it is rather the quality of the DNA which is important. So we also compared quality based on fluorescence of the extracted DNA between the Miniprep and the silicon membrane. And the silicon membrane provides slightly better DNA. As mentioned before, impurities are more common in the Miniprep. And here, the age is correlated with DNA quality. Cates et al. Found that the age is also not correlated with yield, but in fact with the on-target sequences in the HYPE-SEQ, supporting our analysis from Veronica only. And one recommendation was that definitely not sonicate DNA from herbarium specimens further. So why is the quality of the Miniprep lower? It seems, based on different absorption, that the lower amount of phenols and proteins in the Miniprep extraction should actually increase the quality of the Miniprep extraction. However, the Miniprep extraction method is much poorer in removing polysaccharides from the DNA extraction. So this seems to be especially noteworthy when you have problems with polysaccharides in your plant material. An additional factor is the retention of smaller DNA fragments in the Miniprep extractions, which are washed away normally in the silica membranes. So if you're looking for the small DNA fragments and you have less problems with polysaccharides than with phenolic compounds, then actually the Miniprep method may be better for you. However, there are so many other parameters that may influence your results, like the initial amount of plant material used, how you grind them. I mentioned the different lysis buffers, and there are special lysis buffers suggested for ancient DNA. Incubation time can be optimized. As mentioned, the brand of silica membrane may have an effect, the way in which you clean DNA, and there are many other specialized ways for DNA extraction for ancient and herbarium material. Important is to really identify the problem you have in DNA extraction and compare different taxa with your DNA extraction methods. Use material from different herbaria and different ages to find out what is the problem in your DNA extractions. One method which is often suggested for herbarium material and which we tried is paramagnetic bead cleanup, SPRI, which works by binding the DNA reversibly to some beads which are magnetic and can then be kept inside the tube while the DNA extract is washed and many other compounds are washed out. And after removing the magnet, you can elute the DNA again. This is considered a very efficient cleaning method. However, in our experiments, it received a lot lower yield than the normal method. So we do not consider it really suitable for herbarium material because of breakage of DNA likely happening in the process. However, one thing which I need to mention towards the end is be aware of contaminations because they are really everywhere, and I have experienced the strangest contaminations. Many of these contaminations you cannot really avoid because plants have many endogenous pathogens and symbionts, which you of course extract together with the plant material. However, there's also some fungi and bacteria living on the leaves, which eventually you can wash away at least a bit. And there are many different contaminations in the lab equipment. For example, the most common contamination is through the pipettes. Of course, there may be also contaminations coming through the air during the DNA extraction. So you need to prevent these contaminations, and I gave some recommendations in the slide before you start. Of course, if you work swiftly, you may prevent contaminations, and you should not extract more than 10 plants in parallel when you're working with herbarium material. But it is also necessary to check for contaminations afterwards. So use negative controls in your DNA extract, blast sequences afterwards, or check against the reference genome. And one study which did this, and which will be presented later, I assume, was the one by Franziska Patzold mentioned before on Lepidoptera. And if you can look at the different samples included in the study, you can see that some have really some high percentage of human contaminations and also some unclassified contaminations. There are even other studies who have shown that in many cases, people have sequenced and used in analysis rather the endophytic fungi rather than the plant itself. So try to avoid these. There are now good genomes available for a plant relatively close related to yours, I'm sure, and that can be used to identify those contaminations. The final step, of course, is DNA storage, and one needs to take care of ethanol being completely removed because it's an important PCR inhibitor, storage in water or TE buffer, the latter especially when you plan for long storage, contaminations being prevented during the DNA storage, and storing at -20 or lower. And further, reduce the freeze-thaw cycles by making aliquots. And with that, I thank you for your attention, and I'm open to questions. And of course, I provide here the literature that I used and recommend you for reading. Thank you.